Afternoon, and I'm just going to give a short overview of some of the collaborative work which we've been doing with QMUL, between QMUL and King's, and just give some ideas on artificial intelligence. And thank you to the amazing Helen Morriman for inviting us to talk about some of these ideas. Thank you very much. Where'd I point? Okay, so I think everyone, there's been a lot of AI in the news at the moment. And the main reason for that is because there's been, an explosion in the amount of data, good quality data available for computational modeling. And in parallel, there's been an increase in computing power in terms of high performance computing, and also the development of neural chips, which facilitate the training of AI models. And in addition, over the past decade or so, there's been increasing refinements in the algorithms and also in the software infrastructure, which facilitates training of AI. So what is AI, or also known as machine learning? It's the way in which we enable or ask computers to learn from data. So basically what the models involve is you put your data into the system. The data has to be all pre-processed in that it has to be cleaned up and, dimension reduced, and then feature selection. And then you put it into whatever algorithm it is, and then you get your prediction at the end. And, there's different layers of algorithmic complexity. Which way does that work? Oh, there. And with increasing complexity, so starting off with fairly standard ones, which support vector machines, random forests, or gradient boost, and then, as I said, getting more complex, going into the convolutional neural network. So this is another representation of a machine learning pipeline. So you have the different layers of data, and in this scenario, it's genes, proteins, and metabolites. You've gone for a machine learning approach. It can either be an unsupervised learning, which is where the data is not labeled. So then you use a clustering approach. So I think, you did principal component analysis, and you've been seeing it with a single cell analysis data, where they've been representing large data sets in computational space by using TSNA or UMAP, which is a way of representing that sort of data within the latent computer space. The other way is supervised learning, is where the data's labeled. So you can either do a classification system, which is, is this cancer, yes or no, or a regression system where you've got a point and you want to predict where that point is on the line, and that's classically done in survival. So as the systems get more complex, so you have your input layer, and then you start going into your hidden layers, where you have multiple layers of nodes, which can vary in the number of nodes in each layer, and then you get your output layer, and depending on what you put in, it will determine to some extent what you get out. All right. This is just showing again sort of, neural network design, how it gets, the complexity of it, and the way in which they can learn. These are artificial neural network, recurrent neural network, or convolutional neural network. And it's whether it's a feed-forward system or a back propagation. And that has some bearing on whether it's going to be, it's on the training efficiency, training speed, and also on the computational time it's going to take. And this is just sort of highlighting that, for the nodes, you can have different mathematical functions which you use to define a way in which you handle your data within sort of computer space in order to model it and work out the answers that you're looking for. So deciding what algorithm you're going to use ultimately depends on what your problem is, what the data is, what you're looking for, speed or accuracy. Is it an explainable, to who you're trying to explain it to, and is it an acceptable solution? So commonly in clinical practice, everyone is happy with using linear or logistic regression because it's understandable, but its accuracy is not so great. But if you start going into deep learning, its accuracy is high, but then you're getting into a scenario of the black box, which is all those hidden nodes. People don't understand it, and they're a bit wary in trusting what the computer is recommending. So why is AI attractive in applying it to cancer. I think everyone in this room knows that cancer is highly heterogeneous, and in particularly cholangiocarcinoma, I think I would say it's one of the most molecularly complex cancers out of all of them. And AI would facilitate, hopefully, a way of identifying markers predictive of response and a way in which to personalize therapy. So some aspects of AI or using machine learning to predict drug responses has been used to a certain degree in terms of using genomic features, gene expression patterns, copy number, or transcriptomic data. And this has been facilitated by a number of open data resources. But there's not so much done on the cancer phosphoproteomic profile. As a cancer evolves, it gets sort of sequential accumulation of multiple kinase pathways being switched on, which is basically, you're getting a phosphorylation process to a protein, and that typically leads on to that pathway being switched on. And there's some evidence that if you look at kinase activity, that has a better predictive power for therapeutic response over genetic mutations. But as I said, that is only in the field of heme-onc. So just to explain how you get your phosphoproteomic data, essentially you digest up your chunk of tissue or your cells and breaking up all the proteins into little bits. And then based on their mass and their charge, you then separate them all up into little bits, and then you end up with your spectral map. And then you apply standard computational mass spec to basically put all the bits together to work out what the bits of, what proteins you have, to quantify it, and also to work out whether they're phosphorylated. And this is the input which is used for the next two algorithms, which I'm explain to you, which were both written by Professor Pedro Cutillas over at QMUL. One of them is called KISEA, which stands for Kinase Substrate Enrichment Analysis. So basically you have your tumor tissue and your background tissue, and you compare their phosphoproteomic profile. And by seeing what phosphopeptides are up, you can then enrich for which kinases are activated, and then you can infer which kinase inhibitors would be effective in blocking those pathways. Now, Drummond's my favorite algorithm. So this stands for drug ranking using machine learning. This is, I'll just explain a bit more about how it was developed. So we got the training data, or the input data, came from PharmacoDatabase, which is a fantastic open source repository of over 400 drug responses in, I think, over 150 different types of cancer cell lines. And then we combined it, and it documents the drug response according to the anti-proliferative response. We then combined that with in-house phosphopeptide data on both solid and non-solid tumors, and there was a bit of transcriptomic data, which was downloaded from a public repository. So that input data set was then divided into the training data set, 80%, and then 20% was just put to one side to be later tested. And for our validation data, we downloaded that from PRIDE, which is a fantastic, again, another open data resource, proteomic identification database, and downloaded the phosphoproteomic and proteomic profiles from a variety of different solid cancer cells, which were not the same as these two. So the way in which, one of the big first steps with any machine learning model is sort of dimension reduction or feature selection in order to make it manageable. And the way in which this was done was using the median of the cell responses to a given drug to divide it into cells that were resistant and cells which were sensitive, and then do a comparison of the features of resistance and features of sensitivity to get an empirical marker of the drug response. Oops, that's the wrong thing. And that basically was used to calculate the D metric, which essentially is just the difference in the distribution of features of sensitivity versus the features of resistance. And this is a nice way of doing things because you don't need any reference or any way of, you don't need another sample to normalize things. So the D metric was then put into a number of different algorithms or deep learning or machine learning models. which are basically different ways of defining things in computational space to model them. And this it shows one of the outputs for a given cell line, showing the D metric for the different omic data sets, red being sensitive and blue being resistant. And I think you can just see there's more actionable targets coming out from the phosphoproteomic data than that which is revealed in the proteomics or transcriptomic data. So Drummond's performance was then tested on that small set of data, the test data which was kept to one side to see how well it was performing on hidden data. And root mean square error is sort of a standard metric using to assess performance of machine learning models. So the different algorithms were performing reasonably well. And then we looked at how well, Drummond's predictions correlated with that of the validating data set, which was downloaded from PRIDE, and that showed a good correlation. So we've then moved on and we applied both these algorithms to cholangiocarcinoma. So we took a number of different cholangiocarcinoma cell lines, both intrahepatic, extrahepatic, and gallbladder cancers. And then we also had 13 cholangiocarcinomas, again, a mix of intrahepatic and hilar cholangios with associated background tissue. We did phosphoproteomic analysis, and then we put the data into both KICEA and into Drummond. These show these are the patient samples, just showing that the samples cluster according to whether it's tumor or background and whether they're replicates, and similarly for the different cell lines, so showing good technical reproducibility. And also there was a good correlation between the phosphoproteomic signatures from the primary tumors to that of the cholangiocarcinoma. And this is just showing the distribution of where the phosphopeptides were appearing in terms of ontology in your hallmark genes. Again, this is just showing how the samples cluster. So you've got your background, or these are the patient samples. So you've got your background liver all clustering together, your tumor samples clustering down here. And this is just showing how the different cell lines all cluster. I must admit, everyone uses MMNK1 as a benign cell line, although it's sort of hanging out with all the cancer. And for the reference which was used for the KICEA, MCF10A was used, which is a fibrocystic breast cell line. This is just an example of some of the phosphopeptides coming up and the profiles within cell lines, within tumor and background. And I think it's just of interest to just see how much phosphorylation events are happening in cell lines compared to what actually happens in tumors. So this is the output from KICEA. So this is just showing the kinase activities identified in the different cell lines and the kinase activities identified within the different tumors. And these are heat maps and hierarchical clusterings in both the cell lines and the tumor. I think you can just see how it's quite chaotic, and it's all, it's not, there's a lot of things switched on in different ways. These are some selected markers of kinase activities, background tumor tissue and cell lines. And we tested a couple of kinase inhibitors across a number of different cholangiocarcinoma cell lines individually. On their own, they're not particularly potent. So we tried some combination therapies which increased the potency, and this was just in one cholangiocarcinoma cell line, which was an extrahepatic one, EGI1. So this is the output from Drummond. This shows the top 30 drugs in the cell lines and the top 30 drugs in the tumor cell lines. In the resected tumors. And again, there's a good correlation between the predictions of the different drugs in both tumor and cell lines. As we had the background tissue, what we also did was apply Drummond to the background liver. so we could then sort of select drugs which would have a higher predicted therapeutic efficacy in the tumor over the liver, just exploring whether it would have any utility in exploring the side effects from drug therapies. And this is the output from Drummel, showing the different rankings of the drugs for each individual patient. And I think it just shows how much variability you get in the ranking of the drugs for each patient. So for further in silico validation of Drummel, we took the predictions which it calculated from the phosphoproteomic data of these different cell lines, and then cross-referenced that with PharmacoDatabase, and there was a good correlation. And then we tested in a couple of the high-ranking drugs, which are a couple of HDAC inhibitors. We also tested response, and we took a low-ranking kinase inhibitor to see how that was performing. Now, Drummel's an algorithm early in its development. I think it's just showing the utility of using AI machine learning in working out what drugs may be of higher benefit in each individual patient. But one of the limitations, I think, at the moment is, PharmacoDatabase doesn't actually have any drugs which are in clinical use for cholangiocarcinoma. So, so it needs more data, basically. So, as I mentioned, what next? As I said, it's more data. And also, I think what's really important is well-curated and well-labeled data. So you can build up these layers of omic data for each individual patient, where you also have the clinical data in terms of what treatment do patients go on, how do they respond, do they survive, and then you can start making prediction, and then you can use that for training your algorithms to say, what drug's going to work in each patient. And ultimately, all of this data should be open access. So and in the data compute with computer people, the most important thing is making it findable, accessible, interoperable, and reusable. And just to highlight, by making a well-labeled data set available and open source that everyone can access it and model it, this led to sort of a big jump in AI image recognition. Ultimately, this all depends on patients, because all patients need to, get involved. They need to give consent for their data to be shared, and also importantly, their biological data, whether that be blood samples or tissue, in order to move things forward. And finally, this is just to thank, I'm sort of more the storyteller. Lots and lots of people were involved. This is the QMUL team, and this is the King's team. That's Pedro. And of course, Prof. Heaton, who got on his bike, raised a lot of money and said, let's do some research on cholangiocarcinoma. So that's probably the main reason why I've ended up here. Anyway, so I hope you found that useful.
Thank you very much. Thank you. So we've got a few minutes for questions. If you just stay at the podium, or you may have a seat, it's fine. Either way. Chiara.
Someone who doesn't know anything about AI. In your last slide, you showed that we can integrate different sets and types of data. My question is, how is important that those different sets come all from the same patients, or how good is AI to actually integrate different cohorts of patients?
Well, it's perhaps a lot of it's on statistical modeling. So so you actually get the power of the algorithm comes from, having a good number of cases. And then you train it and, with all the complete data sets. And then once it's trained and it's working all right, then you can use it on the individual patient. And maybe, and hopefully in the future, you won't need all the layers of data in order to get the answer that you're looking for, because you've already trained the algorithm from the main labeled. Oh, right, thank you. The main labeled data, if that makes sense.
But if you want to create a multi-layer prediction, you need to have the different sets of data coming from the same patients.
Yes. Yeah.
You can't use different cohorts, one for proteomics, one for
No,
gene
no,
expression
I
analysis and
personally-
make single prediction and combine them together?
No, I personally think, for developing the model at the beginning, it should be all from an individual patient, all the multiple layers. I know in a practical point, in practice, that's probably not going to happen. So you're probably going to end up just getting certain layers for each patient. And then from that, it sort of ends up being a jigsaw, and then you build the model, is sort of what I'm thinking about.
I hope you'll forgive me for asking another naive question, a bit like Chiara. This blows my mind. I never quite really capture all of this. Are we, as a community, better having many more patients included in terms of numbers with a small amount of information? Or are we better focusing on a smaller number of patients who are really well annotated in terms of histology, radiology, treatment, outcomes? Which is the best approach?
For me, I think it'd be, to begin, it would be an extremely well curated, smaller database. And I didn't mention radiomic data is another amazing layer of data. And I must admit, it's interesting to see what AI is doing with that at the moment. So I would just say, you don't need to get every patient, because we're still at the stage of, refining and developing the models, which will be for, hopefully, and then get the evidence that, that these should be used in clinical practice.
Yeah. Unless somebody else comes to the microphone, I'll keep a couple more, one comment and one question. You're absolutely right about radiomics. So we do a lot of scans on patients at diagnosis, and we very much use it thereon after in a binary yes/no. Cancer better, cancer worse, carry on. Cancer better, cancer worse, carry on. I don't think we're using all of the information that is available in the scans, and that's such a rich resource for imaging. How can we as a community move on from this?
Well, I think, there's a number of AI centres developing, throughout the UK. There's also an AI division of NHS, which is NHS Transform. It used to be NHSX. So they're already, the infrastructural spine to the UK is getting developed for that. And they're trying, they're aiming to create these trusted research environments where you put patient data for, modelling and answering these sort of questions. The radiomic data is, it's not my field, but it is interesting what's happening there, because basically it's a computational representation of the human body, and then you're asking, the computer again to see what it sees, and it's taking the data from it in a different way in which we see it, which is an image. Like I don't know whether it's come out yet. There's going to be a paper in the Lancet, which is basically they asked the computer whether it could tell the gender from CT scans, which it can apparently very well. And even that's when the the computer will see things in a different way. That's the reason why it starts getting to the black box. It gets into an area where we don't always understand or able to explain how that data is modeled within computational latent space in order to get the answer which you want at the end.
And if I may, just one final question. You said the drug prioritization tool selects drugs based on response. Does that mean hitting the target? And the reason I'm asking that is because one of the new drugs that we're going to be using for cholangiocarcinoma, ivosidenib, we know improves overall survival, but you don't get a great response. It's more of a static drug. So would that drug effectively not be prioritized because we're looking specifically for a response, or is it more about hitting the target?
Because Drummel's, was trained on the data from Pharmaco database. That's just based on proliferative responses, okay. and that's it. So that's the reason why, I was saying, I think the D-metric's a great way of reducing the data and rationalizing it, because it gets rid of noise and you don't need to normalize. But as I said, it's the input training data, or the data which was to build it, needs to be expanded upon. So, deciding what other different cancer endpoints are useful in order to train it to predict response.
Thank you very much.